And from there, we have an evolution of these pigment types and these different chloroplasts or chloroplasts that contain different types of pigments. And by looking at different pigments and presence of different pigments in different cells, different types of organisms, we can actually trace the evolution of that. So by using pigments, we can differentiate different taxa, no? Very simple, awesome, we have a paper, no? Well, people have been using these pigments in taxonomical tools. Some of you have heard maybe about ChemTax. ChemTax is something that everybody does when you usually start your PhD. You go and you take all your HPLC data, you do ChemTax, and then your advisor comes and says, like, no, you should not do ChemTax because ChemTax has to be trained and so on. Going back, all of you had a plant. All of you killed a plant. Probably you killed it because it didn't have enough light. Next time you buy a basil plant or something, put it next to light, put it next to window, let it be there for 10 days, and then take it in the dark and let it be there for 10 days. You're going to see how different green is going to become. The ratios of pigments within a cell that ChemTax is based on are not just defined by evolutionary origin, by their taxonomy, but they're also highly defined by the nutrient conditions they're in, light conditions, and some other stuff. Every time you do ChemTax, you have to train it with a data set that is local to your area that you're working on. No? ChemTax is also limited because it's really looking at only like around 20, 25 pigments, which is nothing. There's new techniques, new clustering techniques that are actually looking at the whole diversity of 124 pigments. This is amazing. I kicked out a slide, but they had 34 cells, 34 different algae, and they analyzed 124 pigments. And they find an amazing diversity. Actually, within the Alexandrium, who was Alexandrium? Emmanuel and a couple other people. Only just within Alexandrium tamarense, they found a pigment way to distinguish the toxic type from non-toxic type. So you can do many different things, but the question is, how much are you willing to pay, and are you going to be capable to trace the differentiation using your optical tools or from space? All right, this is the last one, function or role. Okay, so we come to the thing that has the worst rep in ocean optics since the case 1, case 2 water fights. Never mind. So when you say phytoplankton functional type, you're going to have me like twitching and quite a lot of us, because it turned out to be kind of throw it all, like just kind of like something where you can dump anything what you want the way you want to group the phytoplankton. In theory, like, I'm a classical taxonomist. If you want to group the phytoplankton, use taxonomy, and that's it. Pigment composition, duh, but it's really not clear enough. Use microscopy, use DNA, stop it. But PFTs, these phytoplankton functional types, is more like of a, I would like to group my phytoplankton kind of based on the ones that can do nitrogen fixing, and some. You can group it whichever way possible, and this definition kind of start allowing a bunch of these different algorithms from space to develop. They were kind of like grasping the fact that some theoretical ideas about different types of phytoplankton, use these theoretical ideas to develop algorithms. When you use theory to develop algorithms, for example, the things that like pink and things that don't like pink, it's really hard to find your validation data sets to validate that. And that's where things start falling apart. So over last 10 years, there was an explosion of phytoplankton functional type algorithms that are sadly mostly unvalidated. And this is why this name got a bad rep. So what we're going to do, we're going to call these phytoplankton groups, okay? And now we're talking about awesome thing. There's phytoplankton groups, they don't have a bad rep. These are different ways that you want to structure phytoplankton. You can use taxonomy, you can use pigments, you can use everything, but you can also talk about function, because knowledge about its functionality in ecosystem can help you a lot to discern them from space, okay? So when you talk about the function, we talked about their pigment composition, we talked about their size, we talked about taxonomy. So we're talking about their function. What do they do? Some of them export carbon, some of them don't. Some of them have silica, in them. So therefore, they're going to be appearing in the water only with their silica, because there's no way for them to live in the middle of the South Pacific, because there's no silica there. If some of them need lots of iron, they're going to be in the area of lots of iron. So these are kind of little functionality of the things that you have to remember. They have a function to be very, very nutritional for the hydrotrophic organisms. There's a bunch of things. Some of them have ability or likeness to live in turbulent stratified environment. This functionality can come in really helping when you try to think how to measure these phytoplankton groups from space. There's only so much that you can squeeze out of the radiances, only so much that you can squeeze out of the optics. So if you know the diatoms are never going to show up in areas where there's no silica, don't look for them there. Just put a filter on your algorithm that does not look for diatoms in the middle of South Pacific because there's no silica there. If I told you previously that coccolithophores like high amount of light, they're not going to be blooming in the middle of the winter in North Atlantic, no? So try to think about these ecosystem traits, this functionality, to use it as a filtering condition to build your ways to see them from space, no? How many of you are working on seeing phytoplankton groups from different things for your PhD? Okay, well, yeah, you too. You? Who else? I mean, that she does. 1, 2. Okay, this is Laura. 3. Yeah, you 2. Yeah, 4. Yeah, okay. It's not bad. It's okay. It's okay. That's why you're here. The popularity of this, you know, we've been looking at Earth for the last 20 years from space, no? And we're seeing these pulses, and we're seeing this green, and it allows us to understand so much more about the functionality of the ecosystems. But now we want to know more, and of course, you want to know who's there, because it makes a big difference, as I said previously, if it's a dinoflagellate bloom or is a diatom bloom. Dinoflagellate bloom, it's not going to be so yummy for zooplankton, but diatom bloom, oh, yum, yum, yum. And if you want to understand how much of that carbon gets exported, you want to know who's there. It's really important. So this like fashion about, you know, knowing which phytoplankton are there start increasing. So if you look at for the Google searches, no, they start increasing as we're launching new satellites, and this is very US centric. I apologize for that. But also with the, you know, in situ development of, you know, of different instrumentation, we start more and more people Googling that and trying to work with that. But so how can you study these phytoplankton, you know, with optics? And this is what you're going to be learning throughout this next 4 weeks. You know, once again, we talk about them as size, but then morphology. You can focus on specific morphological and structural features that impact the light. They have specific pigments associated with evolution. No, the specific pigments can help you discern certain things. They have specific size and morphology that's going to leave for the specific optical signal. Think about coccolithophores. No, it's calcium carbonate, very like intricate stuff. They scatter light. You can see them from space. But also think about these taxa-specific ecological traits and trophic states. Think about, you know, what they like, what they want, where do they live. We have 350 years of knowledge about this phytoplankton. Taxonomic knowledge and ecological knowledge sees that. As Emmanuel said yesterday to me, don't be blind, use it. It's, you know, it helps you out. It helps to focus on, because, you know, optics can do only so much. How much? This is a slide for a colleague of mine. He's Ocean Optics class 2011. So I'm going to show you the optics of 2 completely different species. This is a centric diatom, present everywhere except for the polar system. So again, I said this is a really big diatom, has silica, it's very nice, has different completely pigments than this rapidophyte. Rapidophyte is they're teeny tiny, let's say, very looks similar to, like, say, dinoflagellate, but not really. They have flagella. It's a harmful alga species. It's a mixotrophic. This one is autotrophic. High genetic diversity. Completely different thing genetically. And you think about, okay, they should be, you know, kind of different, no? So you can start measuring them in a culture. So this is your absorption spectra for both of them. This is, it's kind of standardized, no? So it's just normalized for 650, so you can compare the shape. Shape looks pretty similar, no? I mean, this is 10 micron, this is, if 10 micron, this is like 60 micron size, no? Completely different pigment structure. Shape looks similar. You can see this blue bump here. So this is your blue is heterosigma, red is your diatom. There's a little bit bump here, probably some helper pigments. There's bumps different, but the shape looks very similar, no? Your backscattering. This is your backscattering slope as a function of, so backscattering is a function of light. No, so backscattering is dependent on their size, dependent on morphological characteristics. Looks a little bit very similar, no? Probably this blue slope is a little bit higher because heterosigma is a little bit smaller than the diatom, but pretty close by. If you look at their particle size distribution, in a culture, you can see this is your heterosigma bump, this is your diatom bump, so they're smaller than this diatom. So what you can do is take this data, plug it in a hydrolyte, and you're going to let guys learn about it and calculate remote sensor reflectance, because it's a completely different species, and you get pretty much same answer. Go ahead.
What are the units on the PSD for reflexes?
This is meter per micrometer, and this is micrometer. So you plug this stuff into hydrolyte, and guys, you're gonna learn about the hydrolyte. Is the magical tool that makes you model the remote sensor reflectance. You pretty much get a very similar spectrum for two completely different species. So what are you gonna do here? Well, you're gonna remember this guy likes coastal warm waters. No, this guy is happening everywhere around the world. Is a global, but it's a silica. You need silica, and usually going to bloom in spring. So you're going to start bringing these environmental things, the traits that we know, the things that we have collected for the last 350 years, to start distinguishing these things. But don't think that optics is going to be enough and hyperspectral is going to be enough, okay? So throughout this whole week, 4 weeks, you're going to be hopefully able to understand by yourself how feasible is actually to study phytoplankton groups with optics, no? And you're going to be able to answer that question towards the end. Remember, know who are your friends, who are your enemies, and that means from methodology-wise. Be realistic on your expectation, and only develop the algorithms that can be validated. Because if I get your paper, it's not going to look nice, and this is not a threat. I'm been kicked out of every PFT working group and known to humankind, same as Emmanuel. For the same reason, because we're asking for validation. So just think about this stuff. You have fantastic tools, fantastic things in your hand, knowledge of 350 years, amazing technology to distinguish different phytoplankton types, sees everything, and really use it to develop new ways to see phytoplankton for space, because they're awesome. And that's it. I'm done. I'm open to any questions. We're going to take a break. Any questions? The slides are going to be posted. I can talk about phytoplankton for 60 million thousand years in so many different ways. And it's really hard to get you up, everybody on the same scale. If anybody who has never heard about phytoplankton before wants to talk on the side, we can go and just chat about it. You're not going to be doing so much phytoplankton cell, but you're lucky that there's an IFCB, so inline flow, imaging flow cytometer here. So you're going to be able to collect samples and actually run into this and see these things that you're going to be measuring pigments and absorption, scattering, with your eyes, and that kind of like allows you to see this diversity. But yeah, phytoplankton are awesome. Just don't be mean to them. No. Don't try to develop bad algorithms. And again, this week, I'm just going to give you a little introduction. You're going to be hearing a lot more about phytoplankton throughout the stuff. Jeremy, he's going to come and talk a little bit more about phytoplankton groups from space in a week 3. Okay? Break? Pee-pee break? Good. Don't. Again, awesome images in diatom.org. Look at this.
Do you know if there's been any algorithms developed that map phycoerythrin?
Phycoerythrin? So there is papers that have tried to target phycoerythrin. I can show you some of that. I mean, this is from a perspective, what would you like to target? Just phycoerythrin or just specific group, or?
I'm not sure about specific groups. But
yeah.
Like I've been looking at phycocyanin, and like studies like I'm looking at right now is brackish water.
Yeah.
And I figured maybe it's more reddish than it is
yeah.
blue green. So
So ask me, and then I can